by which these restriction endonucleases break the DNA molecule. Some restriction endonucleases cut the DNA molecule a little away from the palindrome sequence. Therefore, this results in the formation of what are known as sticky ends. Whereas there are restriction endonucleases that cut the DNA molecule exactly at the center of the palindrome sequence. Exactly at the center of the palindrome sequence. This results in the formation of what are known as blunt ends. So, sticky ends and blunt ends. For example, restriction enzyme EcoR1 recognizes the palindrome sequence GAA TCC. So, from the other end also it is GAA TTC. This is the 5 prime 3 prime end. This is the 3 prime 5 prime end. EcoR1 cuts the DNA strand. This restriction endonuclease EcoR1 cuts one strand at this end, the other strand exactly at the same sequence in the opposite direction. So, it leaves a sticky end on each side. So, these are called sticky ends. The advantage of these sticky ends is that they can be joined with similar sticky ends of the other DNA molecule using the enzyme DNA ligase. Another example would be for the restriction enzyme HIND3 cuts the DNA at a palindrome sequence which reads like this: from 5' end AA GC TT. So in the opposite strand it will be AA GC TT, that is the palindrome sequence. So HIND3 cuts the DNA at this point, so leaves a sticky end. So such ends are called sticky ends, and as I told you, sticky ends are useful in joining this DNA with the other DNA using the enzyme DNA ligase. Whereas some of the restriction endonucleases result in the formation of blunt ends. Let's take a few examples for this also. One example is restriction enzyme ALU1. ALU1. This restriction enzyme is obtained from the bacterium Orthobacter lutens. So ALU, first one. This cuts the DNA molecule having a palindrome sequence like this: AGCT, AGCT. This enzyme cuts exactly in the center of the sequence. Therefore, the end will be blunt ends. Similarly, another enzyme HAE3, another restriction enzyme HAE3. This is obtained from the bacterium Haemophilus aegypticus, HAE, sequence number 3. This restriction enzyme recognizes the palindrome sequence GGCC, and the enzyme cuts exactly in the center at this point. Therefore, it will be again blunt ends. I was telling you that when you get sticky ends, it is easier because the DNA with similar sticky ends can be joined to this. In case the restriction enzyme breaks the DNA molecule into pieces having blunt ends, then it is necessary that in such cases a few nucleotides are to be added before another DNA is attached to it. So adding a few more DNA nitrogen bases, this is called extending tails. Extending tails.